I am Jennifer Chagoya, and I am a technician under the Extension Plant Pathologist here at the Texas A&M AgriLife Research and Extension Center in Lubbock, Dr. Cecilia Monclova Santana. So unlike other production regions where foliar diseases are more prevalent, pod rot is the most limiting disease for producers here in the dry climate of West Texas, affecting not only yield, but also really affecting quality. So we have received reports from area growers and crop consultants that these pathogens are no longer responding to applied fungicides as they once were. Pod rot is a disease complex caused by various pathogens, including the oomycete, Pythium species, and the fungus Rhizoctonia solani, as well as sometimes Fusarium species. The objective of this study was to test in vitro fungicide sensitivity of Pythium and Rhizoctonia isolates from pod rot samples and use this information to help develop future fungicide application recommendations. So we collected pod rot samples from 4 counties in West Texas: Terry, Comanche, Cochran, and Gaines counties. This was a much smaller sampling than we had planned, but then the pandemic happened and we were shut down. A lot of county agents were not in as close communication with us as usual. And then also it was a pretty dry year. So we didn't see high prevalence of pod rot. For the samples that we did get upon receipt, they were washed thoroughly with distilled water and allowed to dry in a biosafety cabinet. And then the samples were plated on the Pythium selective medium, modified pimericin vancomycin medium, and then everything else was plated on water agar for the isolation of Rhizoctonia. Isolates were maintained and tested on cornmeal agar for Pythium and potato dextrose agar for Rhizoctonia. Initial identification was made by observation under light microscopy, and then identified to species level for Pythium and anastomosis group for Rhizoctonia by Sanger sequencing of the internal transcribed spacer region. So from our very limited number of samples, we recovered 3 Pythium and 3 Rhizoctonia isolates. For Pythium, each isolate represented a different species: ultimum, irregulare, and the non-pathogenic oligandrum. For Rhizoctonia, the first isolate belonged to anastomosis group anastomosis group 4, homogeneous group 1, and then the other 2 belong to the binucleate anastomosis group F. Also isolated but not screened for fungicide sensitivity were Fusarium solani and Fusarium oxysporum. So here are the fungicides that we tested in vitro and the diseases that they target. First is the quinone outside inhibitor, azoxystrobin, which is very commonly used for a variety of diseases. Specifically, it is labeled for the control of Rhizoctonia pod rot and suppression of Pythium pod rot. Azoxystrobin was tested both with and without the addition of SHAM, which inhibits an alternative oxidative pathway that can occur in vitro with QOI fungicides, so that our results are more comparable to what would happen in vivo conditions. Next are the phenylamides, mefenoxam and metalaxyl. Mefenoxam was tested in the form of Ridomil Gold SL, and it is used for the control of Pythium pod rot. And then metalaxyl in the form of the seed treatment metalaxyl 4.0 ST, which is for Pythium seedling diseases, not specifically pod rot. Last is the SDHI, pydiflumetofen, which is in the form of Miravis. And this is not effective against pod rot, but it is commonly used in a fungicide program. So for each fungicide, plates contained 10-fold dilutions from 100 down to 0.01 ppm of the active ingredient, as well as no fungicide controls, and they were inoculated with a mycelial plug and incubated at 25 degrees Celsius. Mycelial growth diameter was measured daily until mycelia covered the entirety of the no fungicide control plate, and then the last measurement before plate overgrowth in the no fungicide control was used to calculate growth inhibition as a percent. This measurement for Pythium ultimum and irregulare was 24 hours, and for everything else was 72 hours. Each treatment was performed in triplicate, and then each experiment was repeated. So here are our fungicide inhibition curves, with fungicide concentration increasing along the x-axis and the inhibition of mycelial growth inhibiting along the y-axis. Azoxystrobin is shown in the blue, light blue without the addition of SHAM, and dark blue with the addition of SHAM. The phenylamides, mefenoxam, and metalaxyl are shown in light green and dark green. And the fungicide, which does not target pod rot, is shown in pink. You can see from the plots in the top row that complete or near complete inhibition was achieved for the Pythium isolates, with azoxystrobin, mefenoxam, and metalaxyl at the highest concentration tested of 100 ppm. However, for Pythium 2, the irregulare, we see that the inhibition levels of between the inhibition levels off between 10 and 100 ppm, around 85%. Wheeler et al. Did previously report reduced sensitivity to mefenoxam in Pythium irregulare isolates in 2005. Also, there's a large difference in azoxystrobin between the with and without SHAM treatments. So for Pythium isolate number 3, the non-pathogenic oligandrum, it did not grow in the presence of SHAM at all, even in the no fungicide control, and it was highly sensitive to mefenoxam at all concentrations. As expected, pydiflumetofen had limited activity against any of the Pythium isolates. For Rhizoctonia on the bottom row, azoxystrobin showed decreased activity as compared to the Pythium isolates. The slopes of those curves are a lot less steep. As expected, mefenoxam, metalaxyl, and pydiflumetofen didn't really have any activity, with the exception of Rhizoctonia 1, where we did see some inhibition from that chemical. So from these inhibition curves, we calculated EC50, or the effective concentration for inhibition of mycelial growth, by 50%. And for azoxystrobin without SHAM, Pythium irregulare approached just 50% inhibition at our highest concentration. So that EC50 is just over 100. Again, Wheeler et al. Reported similar values in her paper in 2005. So the Rhizoctonia isolates had higher EC50 values compared to the other two Pythium isolates, but when we had the addition of SHAM, which is what we expect would happen in vivo, those numbers dropped down. For mefenoxam and metalaxyl, you can see that it had no activity against Rhizoctonia. Again, we don't expect it to. And the Pythium irregulare had elevated EC50 values compared to the other Pythium species. And last, the pydiflumetofen. Again, we don't expect it to have any activity against these isolates, but we did see some in the multinucleate Rhizoctonia 1. So this is all great information, but it's very difficult to translate this to what is happening in the field and those reports that we're getting where these fungicides aren't working like they used to. So at 100 ppm, that's not going to happen in the field. So maybe after application, you might be in the 1 to 10 ppm range. Honestly, after direct application, your values are going to decrease. So we're going to look at the 1 and 10 ppms here. So at the top is 1, the bottom is 10. We have our Pythium isolates on the left side and Rhizoctonia on the right side. And we're going to mostly focus on azoxystrobin in the blue, and then the mefenoxam in the light green, because these are what are actually used for pod rot control. So for the azoxystrobin, what we can see here is significant differences. So ideally, we're going to shoot for 100% inhibition if we can. That would mean we're completely killing the fungus, and not allowing it to continue to replicate. So in the red boxes on the top, you can see that none of the azoxystrobin treatments reach 100% suppression. When we increase to 10 ppm, we do see suppression in the ultimum and the oligandrum Pythium isolates, but the irregulare, we still don't. And this agrees with what we saw in our other growth curves and EC50 values, where that one is a little more resistant. For the Rhizoctonia, we are seeing much lower levels than we would expect, especially since azoxystrobin is actually labeled to control Rhizoctonia pod rot. And then if we look at the mefenoxam, just in Pythium, it's not labeled for Rhizoctonia. Generally, this has better control compared to the azoxystrobin, but we still see some reduced sensitivity in the Pythium irregulare at 1 ppm. So in summary, our results suggest that there is the initial development of resistance to the most used fungicides for pod rot control. Again, this is a very, very limited study, but it corresponds with what growers are seeing and what has previously been reported. So the sensitivity to the fungicides was reduced at concentrations that might be comparable to field applications. Again, it's really hard to extrapolate from the lab to the field. But what we are doing now is taking this information and trying to test different fungicide regimes by combining different modes of action for hopefully the best control of these pod rot pathogens in the field. So we would like to acknowledge the National Peanut Board and Texas Peanut Producers Board for funding this project. Crop consultant Mark Scott, who was very good about pointing us in the right direction of fields that had incidents of pod rot and bringing us samples. IPM agent Carrie Siders, John Kaysen and Brian Bennett at Texas AgriLife in Stephenville for sending us some samples. Mondi Vanderveer for providing chemicals from Syngenta, and then Dr. Terry Wheeler for advice on this project. And with that, I will take any questions.
Thanks, Jennifer. We've got time for a couple of questions if some come through the chat box.
Okay.
Question Q&A part. Let's see. No questions yet.
Okay.
Okay. Yeah. I guess what's the most popular of the fungicides in question where there might be issues? What's the most popular one that's used in your area? And I know Texas is really big.
Yeah, the azoxystrobin, growers really like to apply that even as kind of a preventative measure, which we really cannot recommend due to this buildup of fungicide resistance. And then Retamil is very effective against Pythium, but it's very costly.
I see. Okay, thanks.
Thank you.
Yep, thanks for being here today. Thanks a lot.